In this video, I'll provide a quick demo of the top-down assignment workflow in AutoVectis. We'll be starting with an ICR transient stored in the Bruker.d file and process it all the way through the workflow to a top-down sequence map, including the internal fragments. This is the AutoVectis main user interface, and I've already queued up a myoglobin spectrum for processing that contains the results of an ECD fragmentation experiment of the myoglobin. As this is just a demo of the functionality of AutoVectis, I won't go through the settings in this video, but these are covered in other videos and would be covered in user training. But for now, we'll just start the phasing process to produce the absorption mode spectrum by clicking this Begin button. As part of the phase correction process, AutoVectis needs to identify peaks that will be used for phase calibration, and that's what you're selecting in this window. However, AutoVectis will attempt to automatically optimize the settings, and it does a pretty good job of this. So here, we're just going to accept what AutoVectis has recommended. As you've just seen, the phase correction process itself doesn't actually take very long, and the green spectrum you can now see on the screen is the absorption mode spectrum. Now the next thing we need to do is detect the peaks in the absorption mode spectrum, and we do that by clicking here. In Autoseequer, the peak detection tool, you can change the minimum signal to noise threshold for detected peaks, and for this demo I'm going to set it to 2.5. So now we have both the absorption mode spectrum and the peak list, and we're ready to start the assignment process. In AutoVectis, top-down assignment is done using a tool called Autoseeker, and to start that, we just click this button. Autoseeker matches peaks in your spectrum against libraries of predicted peaks for known protein sequences. As this mass spectrum was not well calibrated, I've done an initial assignment pass in Autoseeker using a very broad error range, plus or minus 60 ppm. And now, down in this display, you can see what the mass errors of the assignments are, and it's clear that the main body of assignments follows a specific trend. Now, there are lots of different ways you can do internal recalibration of data in Autoseeker, but the fastest way, and the way you'd normally do it when you're exploring data for the first time, is just to use the cursors on this error plot. You simply drag them to define the recalibration line that you want. You could then go on to do a more formal internal recalibration later, but for this demo I already know that these cursors are in approximately the right place, so I'm just going to accept this recalibration by clicking this button. And now that recalibration has been applied, I can now reassign the data using much tighter error bounds of only plus or minus 2 ppm. Now that's complete, we can jump to view the sequence map for our assignments. The colours on this view of the sequence map indicate the number of times the same fragment has been seen in different charge states, where purple means once, blue means twice, green means 3 times, and so on. And looking at the assigned fragments themselves, it's obvious that we have good sequence coverage up to this methionine. Up to this point, we've been assigning the peaks in the spectrum using a library designed for oxidized myoglobin. So let's see how much better the results look if we use a library without those modifications. Now we've gone from a sequence coverage of 40% to over 80%, and you can see the increase in the number of fragments in the sequence map. And we've only been looking for the terminal fragments so far as well, so let's also look for the internal fragments. Obviously there are many more possible internal fragments than terminal ones, so I've sped this assignment process up because it takes about 5 minutes in practice. You can always follow the progress of the assignments using these progress bars up on top. And there we have our sequence map showing both our terminal and internal fragments. In AutoVectis, it's very easy to manually review the assignments it's made, and you do that by looking at the forward, reverse, and internal assignment tabs. On these pages you can use the cursor keys to move between assignments and manually review the modelled isotopic distributions against the spectrum. As expected, the internal fragments have much lower signal-to-noise ratios than the terminal fragments. Also, you'll note that a large number of the internal fragments have been automatically deselected in this table, and that's because they match the formulae of previously assigned terminal fragments. And those selection-deselection buttons are also open for the user to use if they choose to add or remove assignments that AutoVectis has made automatically. But once you're happy with the assignments, you can then export them in a number of different formats. So one of the options is to an SQLite database file, which can be useful for searching between multiple results, or in this case I'll demonstrate how to export the results to an Excel file. So this file contains information for all the assignments on 3 different tabs. The forward ions, that's the ABC type ions, the reverse ions, that's the XYZ type ions, and the internal fragments all have their own page. And on each page you get information about the ion name, the formula of the ion, the neutral mass, the figure of merit, which is how closely the isotopic distribution matches the theoretical distribution. You then have the exact masses of all the assignments, the ppm mass error of all the assignments, the spectral mass of all the assigned peaks, and the signal-to-noise ratio of all the assigned peaks. Back in Autoseeker, I thought there was one other feature that I ought to demonstrate, and that's Autoseeker's ability to combine results from more than one assignment. In this case, what I'm going to do is process this same myoglobin peak list with both the unoxidized myoglobin library and then the oxidized myoglobin library, so that I can show the combined results at the end and identify the differences between them. We won't need the internal fragments for this, so I'll switch to the Fragment Classes tab and ignore all the internal fragments. Now I need to export these assignments to a database file so that I can load them back up again later to compare with the oxidized results. The SQLite database assignment files have the extension SQASS. So now we need to reassign the dataset using the oxidized myoglobin library. So I'll switch to the main controls tab and load that library up. And now I can save these assignments to their own database file. Now to view both of these results together, I need to jump to the Combine tab. Using this tab, you can reload a list of assignments files and display them all on the same sequence map. So I'll use the Browse buttons to find the files I want to show. And now it's very obvious that using the oxidized myoglobin file, we got good sequence coverage up to this myoglobin and not after it, and from the other end up to this myoglobin and not after it. Whereas in the unoxidized library, we got good sequence coverage across the entire protein. And if you want to use this sequence map in a presentation or a report, you can export it using this checkbox here. This will export the sequence map as an SVG format vector file, and you can then open this vector file up in Inkscape and change it into other formats to suit your needs. So for example, you can export it as a PNG file if you need it as a bitmap type image, or you can export it as a PDF file or other vector formats. And that results in a graphic like this that you can then use for posters, presentations, talks, etc. So just as a quick summary, the different ways you can get data out of Autoseeker are, as you've seen, the Excel file. You can export the sequence map as either a vector or a bitmap format file. You can export all the sequence data to an SQLite database, and you can export the isotopic distributions of any of the assignments that you'd have seen during the manual review process. If you have a particular style of export you need for part of your own workflow, then just get in touch and we'll try and implement it as soon as we can. So thanks for watching this short demonstration of AutoVectis' top-down capabilities. I hope there was something in there that you found interesting, and please do get in touch with us if you have any questions.